The purpose of this tutorial is to explain the gene prioritization pipeline in the KnowEnG system. In this pipeline, users submit a spreadsheet of gene-level transcriptomic or other omics profiles of biological samples. Each sample also has measurements of experimental or observational numerical phenotypes. For example, drug response or categorical phenotypes, such as cancer subtypes or metastatic status. Multiple phenotype measurements can be provided in a separately submitted spreadsheet referred to as the phenotype spreadsheet. This pipeline calculates a score for each gene based on its relevance to a phenotype and reports the top genes related to each phenotype. The results can be explored as a clustergram visualization. Launching the KnowEnG system will take you to the home screen of the platform. We are going to run the gene prioritization pipeline. Click on Start a New Pipeline to begin. On this screen, you can learn about various pipelines and what they do from the Info panel on the right. This Info panel is also available throughout the workflow to provide information relevant for each step. Click on the gene prioritization and then on the Start Pipeline button to begin the workflow. The first thing we do in this step is to choose the appropriate species from the drop-down menu. For this demo, we'll be using human transcriptomic data, so let's keep the default setting of human. Before completing this step, we need to upload our Genomics features file, which will be a transcriptomic spreadsheet. We don't have any preloaded spreadsheets showing up in the table here, so we'll click on the Upload New Data button at the top right of the table. Here you can drag and drop a file or click on Browse. We'll click on Browse and then navigate to the file in our system. The file should immediately appear in the data table with uploading progress info. Once it's done uploading, click on the checkbox to choose it for this pipeline run. Click on the Next button at the bottom right of the screen to move to the next step. Now let's choose a response file. Here, we'll again click on the Upload New Data button. We'll click on Browse and choose the file on our system. For this demonstration, the response file contains cytotoxic responses caused by 24 drugs in various cell lines reported as IC50 values. Once the file is done uploading, click on the checkbox to choose it for this pipeline run. Then click on the Next button to move to the next step. The next step is to select the statistical method we want to use. We'll keep the primary prioritization method at its default value of absolute Pearson correlation. If we were to change any parameters, we can always get back to default settings for everything on the current screen by clicking on Reset Defaults in the upper right-hand corner of the form. Click the Next button to move to the next step. This step in the pipeline setup is where we decide whether to use the knowledge network in our analysis. If we use the knowledge network, we employ an algorithm called progeny. This algorithm allows us to identify genes that are associated with the phenotype, both directly and indirectly through their network neighbors. If we do not choose the knowledge network, only direct associations are evaluated. We'll select Yes. The knowledge network settings will appear below. Depending upon the type of data and species, choose the appropriate knowledge network from the drop-down menu. A default network is chosen by the system based on the species selection at the beginning of the pipeline setup. Keep the amount of network influence in the analysis at the default value. We will also keep the default number for response-correlated genes. Now we'll click Next to move to the next screen. This step will ask you, do you want to use Bootstrapping? Select Yes, keep the default settings, and click Next. The next page is a summary of parameters that were chosen for this particular run. The system generates a default job name, but we can change this name here. Since the same job can be repeatedly run with only slightly different parameters, you may want to attach a note to the job to help document your process. To access this feature, click on the Notes pencil icon. After we've reviewed our selections, we'll go ahead and click Submit Job. Once the job is submitted, a new page will display the status of the run and should say Running. From this page, you can navigate to the results page or start a new pipeline. Leaving this screen will not cancel your running job. To see your results, click Go to Results page or Results in the main navigation. We'll click on Go to Results page. On the results page, you should now see a listing of all jobs you've run, with the latest one at the top. In our case, we are only seeing the job we have just submitted. Wait for a few minutes until the status column for this entry turns to a green check mark. Then click on the job name and look at the info panel on the right. Here you can access all of the information about this run, including data files used, parameters, notes, and favorite status. From this panel, you are also able to download, delete, or favorite a result. To open the result, double-click on the row in the table or click on the View Results button in the info panel on the right. The main panel shows a grid view of genes as rows and phenotypes as columns, which are 24 drug treatments in this sample dataset. The colored cells indicate an association detected between a gene and a phenotype pair. You can sort this view by right-clicking a specific column to find the top genes for a given drug. In the top left panel, the algorithm provides a score between 1 and 0 for each association. A score of 1 indicates a strong association, and a score of 0 indicates a weak or non-existent association. You can also filter the results by sliding the number of top genes per phenotype. Moving the slider to the left shows only the strongest genes for each phenotype. As the slider moves to the right, the filter is relaxed and more genes are shown for each phenotype. You can also select for individual phenotypic responses. In the main panel, if you click on a colored cell, you will get additional information about that gene phenotype pair. To download the results, click on Results at the top of the page. Click on the latest job and then click the download icon in the right panel to download the zip archive. In the downloaded archive, you will see several TSV or tab-separated values text files. Each file corresponds to a phenotype that lists the genes sorted by their relevance to the phenotype, along with numerical scores for each gene. There is also a file that has such sorted lists of relevant genes for every phenotype. It is a TSV file called Top Genes per Phenotype. Currently, the genes are listed in this file by their ensemble gene IDs, denoted by ENSG. You can look up human-readable gene names by using the genenamemapping.tsv file. This demo was created using a beta version of the platform. The website display and URL may not exactly reflect what you see on your screen. We hope this tutorial has been helpful and that KnowEnG is a useful tool for your research. This tutorial only contained one set of choices for the gene prioritization pipeline. We suggest you try changing various parameters to see their effects. More information on these options is provided on the info panel on each page of the pipeline setup. Additional information about the KnowEnG platform is available in the support section of the platform and on our website, knoweng.org. Thank you.